We talk of precision medicine. Let's shift to precision mitomedicine, AI engineered mitochondrial therapies for this episode. Welcome to MitoGenesis, where we journey through the most fundamental powerhouse of life, mitochondria, and uncover how they shape diseases, healing, and now the future of medicine. When I started working on mitochondria slightly more than a decade ago, therapies were vague, supportive at best, mostly symptomatic. Part of my slow progress was obviously a return to some labs in India with reagents and other issues. But part of it was that AI wasn't there yet. We didn't have all the tools. Now the progress of the field seems like science fiction. The lines between science fiction and real science are dissolving. We are entering the age of precision mitomedicine, where AI, stem cell biology, gene editing, and epigenomics converge to craft custom tailored therapies for each individual's mitochondrial fingerprint. Imagine a world where your own organoids, mini organs, made from your own cells, are being used to simulate treatments, where AI predicts exactly how your mitochondrial DNA mutations will affect cellular energy and recommends CRISPR based edits or mitochondrial specific drugs. This isn't a far off future. It's already taking shape in startup labs and biotech accelerators. To give us a look inside this transformation, we are joined by someone who's building her career rather in just last few years. She's a student who's completed her master's in India and then has been doing research projects in variety of interesting labs, but is showing a lot of potential in regenerative medicine, stem cell biology. Once again, we have Basudha Banerjee. Welcome to the show, Basudha.
Thank you very much, Dr. Khurana. It is great to be here, and you're absolutely right. This is one of the most thrilling times in biomedical innovation. For decades, mitochondria was seen as too complex and too inaccessible. But now we have tools not just to observe, but to engineer mitochondrial health at an unprecedented level. Let's start with organoid modeling, a very, very interesting topic. So, we are using patient derived induced pluripotent stem cells to grow mini brains, mini livers, and mini hearts, each one carrying the person's exact mitochondrial genotype. Isn't that interesting? So, we can now introduce specific mitochondrial DNA mutations, observe real time metabolic and electrophysiological changes, and even simulate aging in these organoids. It's personalized, scalable, and predictive. But that's just one piece.
And I know you know, one can use AI not just for imaging, but for predicting functional consequences. Right, Basudha?
Absolutely. And this is a major leap. So people are training deep learning models on massive data sets. They're combining mitochondrial DNA variants, nuclear encoded mitochondrial protein DNA, metabolomics, and even single cell RNA-seq. So the main goal is to predict how a specific mitochondrial DNA mutation or say fusion fission imbalance would alter the proton gradients, the ROS leakage, or even ATP yield in that context. And these models are already being used to prioritize the drug candidates that target specific mitochondrial complexes, say the complex 1 or complex 5, or even the proteins that are involved in the various dynamics of the mitochondria, for example, the DRP1.
Yep, that's quite interesting. It's incredible. Where does gene editing come into play here, especially considering unique challenges of editing mitochondrial DNA?
Now, that is a very great question. So, mitochondrial gene editing has always been very tricky because of the traditional CRISPR-Cas9 systems that cannot easily enter the mitochondria. But now that we have mito-targeted tools like TALENs, zinc finger nucleases, and even emerging CRISPR variants with custom delivery systems. Some of these use mitochondria penetrating peptides or lipid nanoparticle platforms to ferry gene editors directly into the mitochondrial DNA. Then people are also working on epigenome editing techniques that indirectly modulate mitochondrial function. And that is generally done by altering the nuclear encoded mitochondrial genes, influencing the mitochondrial biogenesis, the fission and the fusion process, and even the buffering capacity of ROS.
So we are not just treating symptoms, but actively rewriting energetic signatures of the cell.
Absolutely. And it goes further. People are even using AI to model optimal mitochondrial states for different cell types. Say, for example, in neurons, there is an aim for high oxidative efficiency and low oxidative species. And in stem cells, flexible glycolysis or the OX4 switching, the AI is helping to customize therapies towards these endpoints. And it also predicts the best timing for interventions, whether it is mitophagy modulation, drug treatment, or even stem cell engraftment, based on mitochondrial signals and epigenetic age markers.
Well, this is the sort of work that will define the next chapter of medicine. It's not just precision oncology anymore. We are talking of precision mitomedicine, using your own cellular powerhouses as both sensors and effectors of health. From stem cell derived organoids and deep phenotyping to AI guided gene therapies, we are reinventing not just how we treat diseases, but how we understand life itself. Thank you for being here and giving us a glimpse of what's coming.
Thank you, Dr. Khurana. I must say that the mitochondria must be ancient, but our ability to harness them is brand new. And the possibilities, they are endless.
Okay. So I'm very glad that you were there. Remember your what powers your cell, what powers your life. This is MitoGenesis signing off, Sukant Khurana, and thank you, Basudha, once again. Thank you, listeners. Bye bye.
Thank you.